let me introduce myself. I am Raghavendran. I'm a research consultant and mentor with Pine Biotech and OmicsLogic. I'm trained in bioinformatics and I hold a computational biology PhD from Max Planck Institute. So my research interest and focus is on data science techniques such as machine learning and deep learning applied to integration of multi-omics data like genomics data, like transcriptomics data, and like epigenomics data, etc. So my role in Pine Biotech includes developing curricula, mentoring students in the research fellowship program, etc. So for my research projects, I leverage a lot of publicly available data to infer very relevant biological research questions. So please let me talk about one such example in a very brief couple of slides, where I developed machine learning models to predict a tumor response for a variety of treatment options, employing multi-omics integration of transcriptomics data and genomics data and copy number variation data. So I don't need to introduce you to the adverse effects of cancer. As we all know, cancer is the leading cause of deaths worldwide. So for such a dreadful disease that we have known for a long time, the systematic cancer treatments have relied on using chemotherapeutic drugs that are basically marginally more toxic to cancer cells than normal cells. This is a huge problem because on an average, only 25% of the cancer patients respond positively to cancer drugs. But in comparison, about 80% of the patients respond positively to popular painkillers that are available in the market. So one prominent solution to this problem is to employ something technically known as precision oncological therapy, where a treatment strategy for cancers designed by targeting molecular characteristics of the patient's tumor. So hence, the treatment is personalized to the patients who are likely to respond to specific cancer therapies. There is not sufficiently enough data from cancer patients and not uniform data from cancer patients to molecularly characterize the tumors that we have under hand under multitude of different treatment options that is being currently studied in different clinical trial levels. So we have to resort to a suitable model systems. So one of the suitable model systems is the immunocompromised mouse model, which is also known as patient-derived xenografts. This is done by transferring the tumor cells from the cancer patients, and before transferring the tumor cells into the mouse models, we subject those tumor cells to next-gen sequencing techniques and gather information regarding genomic data, regarding transcriptomic data, regarding epigenomics data, and regarding copy number variation and other omics that are currently under study. So once these tumor grafts or tumor cells are transferred into mouse models, then these mouse models can be subjected to multiple treatment options or administered several cancer drugs that are currently under clinical trials and study to understand the effect of these drugs and to understand the tumor's response to the respective drugs. So this study actually documents the response of tumor, breast cancer and colorectal cancer and other cancers to multitude of treatment options, and have published this research and this and let the data being available publicly. So this is done by Novartis, and the library and the data that they have deposited and is available freely is called Novartis Patient-Derived Xenografts Library or Encyclopedia. So this encyclopedia or this library has both the molecular characterization data of the tumor and treatment response that are available in the public repositories. So this is a huge amount of data, and so much of data, such a wealth of information in the data generates a lot of research questions, and many research questions have explored this data for specific purposes. So my interest was to use this data to design and implement interpretable machine learning models to predict tumor response to different treatments. So in order to do this, since we have multiple biomarkers or since we have multiple omics data available, so I had to employ a multi-omics integration at a different level. So generally, multi-omics integration can be applied at different levels, like as early as possible or as late as possible, depending upon the complexity and depending upon the depending upon the appropriate strategy that will help us discover complementary information and maximize the information content available in this multiomics data. So for this particular data, I had to include, or I had to integrate it at the late level because of the different distributions of the data along the multiple omics level profiling. So what did we find after using this multiomics data to build a machine learning model? So before the analysis, I will just try to tell the accuracy metrics that I used to validate the prediction accuracy of the machine learning model that trained with this multiomics data and tries to predict the tumor response based on a separate tested data that is separately left over or separated. So the accuracy matrix is Matthews correlation coefficient, which goes from -1 to +1, where the +1 would describe an absolutely perfect prediction accuracy for the model, and -1 would describe a completely negative or completely inaccurate prediction accuracy of the model, and anywhere between 0 is going to describe a random prediction accuracy for the machine learning model that I have developed. So traditionally, for these questions or for understanding the treatment responses, people have looked into the single gene biomarkers that characterizes the onset of cancer, and they have tried to find out whether these single gene biomarkers can also be employed to predict treatment response. But we found out this is not a very accurate model, as you can see from the graph. When I use the traditional single gene biomarkers that characterizes this breast cancer level at a different from different publication for these different treatment options, I see that their prediction accuracies are not very great. But the moment that I include not single gene biomarkers, but information from all of the genes, genomic information from all of the genes, I see that the prediction accuracy slowly start to get better. And I also see that from the precision of the prediction accuracy that is plotted for all the treatment options that is given in this box plot. So the availability of multiomics generally requires, as I mentioned, very complex integration of this data to make sense. So here, increasing the information content by including multiple omics data, we can see that the accuracy of the machine learning model that trained from this data generally increased towards, or everything is going towards +1. And everything has a lot of very good precision in terms of their prediction spread. So this clearly tells us that there is a lot of promise in using this multiomics data to predict the treatment response using these mouse model systems. 1, 2. This also tells us by increasing the information content and by including not a single gene instead of single gene markers, by including many more genomic information and many more multi-omics information, we can increase the accuracy of the machine learning model that predicts the tumor response by learning from the distribution of this machine learning from this multiomics data. And 3, we can probably extend this analysis to a patient cohort or patient information that we can download from TCGA repository, just to test, just to investigate how a model that is developed and that is trained using multiomics data that is generated from mouse models can or respond to patient data from TCGA repository. So what more could be done? I mean, from these results, we obtained a list of informative genes that are currently being investigated for identifying specific biomarker set of genes that can define, or that can be inferred in a mechanistic detail about the tumor response to different treatment levels. So these research projects and these questions can be can be performed by using publicly available data and by using methodologies that are not completely different from what you are going to learn throughout this program and throughout the process of and throughout the lessons that are covered in Genomics platform. So we using the resources in OmicsLogic platform will demonstrate you how to perform such powerful, impactful research and by using or by accessing publicly available data. So as you have been briefed before, the resources in this platform, in the OmicsLogic platform, and in this program, in this genomic program, is aimed at introducing you to omics data and their relevant analysis. And this will eventually help you create or understand or ask a highly impactful biological research question that you can start to explore on your own. So we will demonstrate you over the course of this program how to perform such high quality analysis on genomics data. We will also show you how example projects that we have developed, which start from the first base of formulating a hypothesis or exploring a data, designing appropriate analysis methods, and to perform complex analysis, and in the end, interpret the results in a biologically meaningful way. So as you have all been briefed about the NGS that generate large volume of raw sequence data. So to analyze such a raw data, many different tools for genomic data analysis are used. And these include these include alignment of reads and full sequences, analysis of relationships between these sequences and their evolutionary divergence, measurements of the gene expression and their relationships between sequences, and the relationships between structures of protein that these gene sequences encode to, as well as variations or variants in the gene sequences and the sequences of the genome that can be termed as somatic or germline variants using multiple different calling methods. The Taubot Bioinfo platform is an example of a big data platform that can be used or that can be leveraged to perform all these analysis and many others using an intuitively user-friendly way. So when the analysis is performed using a raw data, which is usually stored in FASTQ file format, there are several important steps to consider, such as pre-processing, mapping, variant calling, annotation, and other associated steps also. So for all these steps and each and every step, we have at our hand a multitude of algorithms that are available, which does basically a similar job, but with a slightly different manner and slightly different protocol. So in this program, we will teach you, or we will help you learn how to use this pipeline and how to use different algorithms that will suit the data or that will suit the question that we have currently in our hand. So for each task, you can connect the analysis jobs into pipeline, and each one of these analysis pipeline will include various inputs and will have various outputs that you can download and use it for downstream analysis. Sometimes sometimes for the outputs, it is just sufficient to visualize the FASTQ alignment files to a reference genome sequence to quickly get a sense of major variants, or to quickly get a sense of how our alignment varies across the reference genome, and what are the different mutant variants that we can observe and that we can find that are distributed along the different genes and different parts of the genome. So this is specifically true, or this is especially true when you have a short sequence or when you have a targeted regions that are sequenced, and these are very easy to explore. But however, to explore a long whole genome sequences and to visualize that and to visually interpret that are extremely difficult. So a computational interpretation and a computational analysis is performed in addition to the visualization that we can use, where this computational analysis can call the differences in the FASTQ read files to the reference genome. These are called variants, and these variants are called from many reads. And in the case of somatic mutations in cancer, that might include downstream annotation of found variants based on the reference database. So these variants can include mutations of different kind, and for each one of these kind, you will have explanation or how to interpret those different mutations and variants that are explained in the following sessions in this program. So to visualize the alignment of the FASTQ files to the reference genome, we will use open source applications, and we will use Taubot Bioinfo platform also to visualize and to learn about their mapping onto the different parts of the genome. So large peaks, this is the reference genome that is in the gray area, and where it has been so far zoomed out that we cannot read the individual nucleotides as you have seen that constitutes this genome. And these gray peaks, large peaks, indicate high coverage in these areas, and shallow peaks will indicate low coverage or low confidence in alignment, as you can see straight away from the gaps in the reads that did not cover these areas. And these vertical lines in purple or in pink lines will tell you what are the differences that we observe at different points in the in our sequence that are aligned to this reference and with respect to the reference. So such visual representation and such visual analysis, as I mentioned, might be sufficient for genomes of small sequences or of targeted sequence experiments. But when you are going to perform such an analysis for 3.2 billion base pairs, it is going to be extremely exhausting and is going to be very heavy in for the computer to also work. So that's why multiple downstream variant calling methods and multiple downstream analysis methods are employed to analytically quantify the variants we observe in our mapped sequence with respect to the reference sequence. So a zoomed in version of these variations or these visualizations often include visualizing in terms of sequence alignment. So these sequence alignments can refer to excuse me, two types of alignment. The one it can refer or it can be aligned to a reference, or which is called a mapping, such as when you have a FASTA files, as I have mentioned, from a sample, and you wish to align each read to the best position on the genome to find whether these FASTA files or whether the sequences in the read or the multiple reads reveal a variation from the particular reference. And in that case, or in another case, multiple sequence alignment, this is when you have multiple consensus sequence that you would like to compare with each other and to develop an understanding of similarity among them or between them. So both of these alignment can also be performed in Turbot Bioinfo platform. In many cases, the multiple sequence alignment can be or is used to find structurally important variation, and such an alignment or such an analysis can also be performed with amino acid protein sequences that can be obtained or that can be elucidated by just simply translating the gene sequences into their respective amino acid coding sequences. This is done by taking the trinucleotide codons, as Elia has mentioned, and then translating them into their respective amino acid sequences. So the differences in both at the genome level and specifically at the amino acid level can often be visualized and can often be identified using or can often be visualized on the three-dimensional structure of the protein that is encoded by the gene, and the location of this mutation on the protein structure and their effect on the function of this protein structure will enable us to make meaningful interpretations of the variants we identified as the result of genomic analysis pipeline. So in another way one can use genomic analysis is by carrying out what usually is called a genome-wide association studies. So to carry out a GWAS or genome-wide association study, researchers use two groups of participants, people with disease being studied and similar to people without the disease. Sometimes this can also include certain variations of the disease or certain conditions that are being studied to identify those genomic areas that are responsible for this altered state or conditions. So researchers then obtain the DNA from each participant, sequence their whole genome, and then if certain genetic variations are found to be significantly more frequent in people with the disease compared to the people without the disease, as you can find in some of these plots here, they can often be associated, or these variations are said to be associated with the disease. These associated genetic variations can serve as a powerful tool or powerful pointers to those regions in human genome where the disease causing problem might reside, or that can be interpreted as very relevant regions that are associated with the disease or the altered state that is currently under study. So the downstream analysis of these found mutations, variations, these SNPs, and often, these can be represented in or presented in a very different way, depending upon the purpose of analysis or depending upon the purpose of the use case. I mean, it can at its simplest form can be represented as a table, and at its more graphical form, it can be represented as a lollipop plot and along the gene sequence, or it can be visualized on the three-dimensional structure of the protein encoded by the gene of interest, and then it can also be visualized as a it can be visualized using multiple different visual forms, and can be visualized along the pathways to which multiple genes that are associated with, and how these different mutations or these different variations in these genes might affect the pathway that involve these genes that are currently under study. So this introductory course leads into multiple courses and projects that can serve as a springboard into the world of bioinformatics for all of you. The curriculum for this introduction to bioinformatics, the curriculum for this genomics course is carefully planned to cover a broad overview of different types of methods to analyze the genomics data, and also to understand how these variety of methods are used to answer very relevant biological questions that are found in research papers involving genomic data and its interpretation. So to learn more about these analysis methods and their applications in various project examples, you will often be referred to associated lessons in the genomics course series and also in other courses in the Learn OmicsLogic platform. These lessons will include more expanded overview of the key concepts, principles, and examples of genomic data analysis. Excuse me. These online courses cover, or these online coursework on our education portal will provide you with introductory materials that have been used multiple times by thousands and thousands of people from all over the world, and they have found these perspectives very exciting. And each of these courses in the end also contains quizzes and also contains assignments that will help you, or that will allow you to see how much you have learned from this course, or throughout your progress through these different courses and different lessons. So in addition to these courses, by seeing how experts analyze their data and how these impactful research results are achieved, you can start developing a research question on your own to identify appropriate type of data and planning and also to design your analysis plan. So for this purpose, we have in our platform carefully prepared some exemplar projects that will use all the skills and all the knowledge that you have learned to infer highly impactful biological relevance and to answer high quality research questions. So these projects include a variety of data, a variety of analysis methods, and ask different questions that are related to using genomics data and and to using multiple different analysis methods to infer high quality biological interpretations. So these practical exercises and exposure to data driven discovery using real research data will encourage a critical thinking for most of you. And the purpose of doing a practical assignment and the purpose of going through these projects that we have developed and then learning along with the projects that we have developed will help you gain a deep understanding of these multiple genomics concepts and the processing and the analysis pipelines involved with it, as well as you can also showcase how various methods of analysis are applied in real world situations. So in the end, by following through these example projects and by also going over the course 9, which will help you in designing your own research projects based on the theme of genomics data, or based on the theme of based on the research area of interest, you can actually perform a project on your own. You can perform a research project, you can answer a research question on your own, and you can have your own research experience that is satisfying for applying various skills that you have learned in this course so far. So that will Yeah, that will conclude my presentation. I will take some questions if you have any, and if you have not, I can hand it over back to Sonalika.